Hello, and welcome back to Biology Explained with me, your host, Mr. Hill. And in today's episode, we are going to be focusing on the processes of genetic engineering, principally that used to get bacteria to produce human proteins. Now, this is part of Key Area 5 of Unit 1 of the National 5 Biology course here in Scotland. It is also applicable to GCSE Science and Biology, and indeed many courses around the world aimed at the 14 to 16-year-old qualifications. So, if you do like this video, please hit that like button and subscribe so you don't miss a future episode. And as always with these revision videos, please do make use of the pause button, okay? So pause when you need to, make a few notes, press play, replay, whatever you need to do to help improve your understanding of this course, you go ahead and do that. Okay, so we're going to begin by looking at the definition of genetic engineering and how it's used in different processes. Okay, so we're going to begin by looking at the term genetic engineering. Now, this term is used to describe any process by which the product is an organism that contains DNA from more than one organism. So think, for example, of genetically modifying plants, where you might have a tomato plant where you would modify so that it grows well in really wet conditions, perhaps. And so you would have taken a gene from an organism that grows in a wet area and put it into the tomato plant, and then that tomato plant is able to then use those genes to then be able to grow in those wet conditions. That is genetic engineering. Likewise, when you take a bacteria cell and you put a human gene in it, and the bacteria produces a human protein, then that bacteria has been genetically engineered. Now, a term we use to describe any organism that has been genetically engineered is the term transgenic. So, for example, that bacteria that produces human insulin, that's a transgenic organism. That tomato plant that grows in wet conditions with a gene inserted into it, that is a transgenic organism. Now, in today's episode, we are going to be focusing on using bacteria to produce human proteins. Okay, this process here. We will look at genetically modified plants in a future episode. In Unit 3, when we look at food production, we will touch on a little bit more detail as to why it is we might want to produce GM plants and the advantages and the disadvantages of using those. But for this episode, we're going to be looking at using bacteria to produce human proteins. Now, in the past, a lot of proteins that were required for medical treatment, such as insulin, which is the example we're going to look at today, would have been taken from pigs. So pig insulin looks very similar, works very similar to human insulin. And so you would take, as a byproduct of the pork production, you would take the pancreas and then you'd be able to extract the insulin. Now, of course, that it has a number of different issues. Okay, first of all, it's expensive to rear a pig to get to the stage of taking the insulin from the pig. But also, it's not actually human insulin, so it's not going to be a perfect fit. And also, many people do not wish to use pork products, either for religious grounds or because they don't agree with it. And so, so when you have a technology where you're able to produce human proteins at a large quantity quickly and safely, then it makes sense to use that process. And that process is genetic engineering. So in our next section then, we are going to be looking at the different stages of this process. So we'll see you very soon. Okay, so for the main section of this episode then, we are going to be looking at how we can genetically engineer bacteria to produce human proteins that we desire. Now, there are a number of different stages that you need to be able to put in the correct sequence here, and there's a number of different keywords to describe the types of genetic material that we're talking about. It's very important when you are describing and explaining this process that you don't miss any steps out. Okay, so it's very common for students to say what happens at the start and what happens at the end of the product, but they miss out the nitty-gritty or some of the specifics along the way, and that's often where you lose marks. Okay, so instead of getting 3 marks out of 3 for a description, you might only be getting 2. So pay attention to each of these steps. I'd advise that you pause the video as you go through it and add to your own flowchart of the different stages, and don't forget to highlight those key words. So we're going to begin then with 2 types of cell. Now, to genetically engineer bacteria, you need, naturally, a bacteria cell. You're also going to need a human cell for which you're going to get the human gene from. But we're going to begin by looking at this bacteria cell here. Now, In that first episode, episode 1 of this unit 1, we talked about the structure of bacteria. And one feature we talked about was the circlets of DNA that you find in these type of cells. You will recall that these little circlets are known as plasmids. And bacteria have these plasmids which contain genes which bacteria actually use to transfer between other bacteria so that they can spread genetic information between themselves. Luckily, as humans, we are able to take advantage of that process and actually take this plasmid out of a bacteria cell and modify it. So the first step in the process of genetic engineering, so step 1, is to extract a plasmid from a bacteria. Okay. So the bacteria plasmid is taken out, and we have our plasmid here. Now, the second step, we're going to go to our human cell. In order for us to genetically engineer bacteria to make human proteins, we need to know the gene for the human protein in question. And this step actually takes quite a lot of research before you get to the industrial stage of making the protein. You have to study the nucleus, you have to study the chromosomes, and you need to understand and work out where the gene and how that gene folds, how that gene is coded to make the folded protein before we get to this step. So there's quite a lot of research involved in this stage. But once you have done so, you need to get yourself that human gene. So in our second step, we need to extract the human gene for the protein in question. We get that, of course, from a nucleus. Okay, so I'm going to draw myself a little chromosome, which we're going to take out of the nucleus, and then we're going to have ourselves a little gene, which is just this section here. Okay, so that is the gene. So we've got the gene here, and we've got our plasmid here, our first 2 steps. Now, what happens next is that we are going to put these 2 things together. So the first thing we're going to do is we are going to cut open our plasmid using enzymes. So our plasmid is going to get chopped up here, and we're going to cut the ends of this with enzymes. Okay, so let me just write this down here. So step 3, enzymes cut open plasmid. Okay, and these are our enzymes here. We've cut open the plasmid. Next step, step 4, is you're going to put the human gene inside the plasmid, like so. And you're going to glue it together with enzymes. So step 4, the human gene inserted into plasmid. Okay, using enzymes. Don't forget to mention the fact that enzymes are going to be gluing these together, just like they are used, different enzymes though, that are used to cut up the plasmid. What we have now here is a plasmid that contains human genes and bacterial genes. Now we can call this recombinant DNA. This is a bonus word, okay? So recombinant DNA is formed when you have DNA from more than one species. Okay, now you don't need to know that for National 5, but you will see it written down a lot, and it's pretty clear what it means, recombine. Okay, so this is when you have combined DNA from more than one species. Right, next we have our complete vector. We're now going to put it back into a bacteria. So you get another bacteria cell, and you're going to insert the plasmid with that human gene into it. Okay, so I've drawn it there, and you can see this plasmid has both species genes. So step 5, okay, the plasmid is inserted into a new bacteria. Okay. Last but not least, this bacteria is then going to be grown and allowed to reproduce en masse. And we're going to do this on an industrial scale inside giant fermenters. The process of growing microorganisms is known as culturing. And so what you're going to do is you're going to take this bacteria and you're going to feed it nutrients, you're going to keep it warm, and you're going to culture it so it reproduces to 2, then 4, then 8, then 16, etc. over a number of days. You're then going to have loads and loads of bacteria with this human gene. Now, because the DNA is the same in any species, the bacteria is going to read that DNA as if it was its own, and it will produce that human gene. It will then secrete it out of the bacteria, and we can then harvest that protein out of the fermenter at a large scale. So in our diagram, what we can do is we are going to continue, and this time we're going to culture our bacteria. So there's loads of bacteria now, each with that plasmid, and it's going to produce lots of human protein. Okay, there's dots here. And that human protein is going to be harvested. Okay, so final step. Step 6. Okay, so bacteria are cultured. And the protein harvested. And there you go, the 6 steps in genetic engineering of bacteria. Remember, this process is quick. It produces human proteins, which are perfect for what we need. And it can be done pretty much anywhere in the world where you can have a sterile, clean laboratory and electricity. As long as you've got a big fermenter, you can grow up this bacteria and produce lots of human protein quickly and safely. Okay, so now that we've had a look at the process, it's time to put all this together and have a go at a question. Okay, so it's question time now for this episode, and today's question comes from the paper 2022, paper 2 for the National 5 biology exam here in Scotland, and it's question 5. Now, give this a read. You'll see that this question has a part of the process that we've just been learning about, and it's been labeled with different letters. This is very typical of a question about genetic engineering. Sometimes it will be a diagram, sometimes it might be a description of each step, but in the wrong order. But almost all the questions are going to ask you to identify the stages or to place them in the correct sequence. So, 4 marks available. Please don't forget your keywords. Have a go and come back in 5 minutes and we'll go through the answers. Best of luck. So, let's have a look at the answers now to this question. So, let's give it a read, as always. So, penicillin was the first antibiotic used to treat bacterial infections. Bacteria have been genetically engineered to produce large quantities of penicillin. This diagram represents some stages of the process. Okay, so we've got a cell A with some genetic material B being extracted and C being taken from that. And then you've got a cell where some genetic material has been extracted and that's been cut and joined together with C to produce D. The question is asking you to use the information from the diagram to complete the table. Okay, so first one, which of these is the bacteria cell? Well, we know the bacteria cells have plasmids, which are circles of DNA, little loops of DNA. And the only cell that has a loop of DNA here is cell E. So the answer must be E. Next, the gene for penicillin. Now, the gene, remember, is what is going to code for our protein we want to make, and it's going to come from the other cell. So if we look back at the other cell, we can see that we have cell A, and the genetic material B is extracted. Now, B is going to be part of a chromosome. The gene itself is just a short sequence of the chromosome, and that's going to code for the protein. So the answer for this one is going to be C. Next, how do we name structure D? Well, we know it's a circlet of DNA, so you're going to get a mark for saying plasmid. Now, it's a modified plasmid because it's got DNA from another species. So if you said modified plasmid, that will get you the mark. If you said plasmid of recombinant DNA, even better. But plasmid is all that you need to know. And that's the only, that's as much as you need to write down to get yourself the mark for that point. Okay, question B, describe the next stage in the production of penicillin, 1 mark. So we're going to take this modified plasmid and we're going to put it into a new bacteria, and then that bacteria is going to be cultured. So the next step is simply just to place it in a bacteria. But it's important that you always say that it's going into a new bacteria. It does not go back into the old bacteria. That is not what happens, okay? It goes into a new bacteria. And so if it's not clear that you're saying new, you won't get the mark. So be crystal clear with that point. So you could say that the modified plasmid is inserted into a new plasmid. Not a new plasmid, a new bacteria. Okay, new bacteria. Make sure it's always that whenever you're answering a question on the next steps of this process. So there we have it, genetic engineering using bacteria to produce human proteins of choice. Now with this topic, it's really important that you remember your sequence. Don't forget the nitty-gritty. Every single step needs to be described and explained. And don't forget those key words, words such as plasmid, genes, chromosome, as well as modified, and where those enzymes are being used. Now, if you like this video, please do subscribe so you don't miss a next episode. And I wish you the best of luck with your revision and hope to see you for our next episode where we'll be looking at the process of respiration. Okay, take care now. Bye-bye.